Hello, doers. Welcome to our third module for the FAIR data training for the deep ocean early career researchers. And we'll be learning today about the best practices in the ocean sciences. And I gotta say, hi, I'm Stace Beaulieu. I'm joining you here from Woods Hole, Massachusetts, USA. But I gotta say, this was the most challenging set of materials to put together. Why? Because this is our domain. It was really hard to pare down the materials so that you have access to just what I think are essential resources. So real go-to resources. So the purpose of the recording today is really to help you to get links to resources that can help you learn more. So I posted into the chat the link to today's slides where you can then find all the other links used in today's presentation. So welcome to those new to the iDOOS project. This is the Implementing a Deep Ocean Observing Strategy project funded by the U.S. National Science Foundation. And DOERs more broadly is an international program. It's endorsed as a UN Ocean Decade program. So I'll be here on the recording, and then our live session will be joined by some seafarers, data specialists in the iDOOS project, including Karen and Leslie, and you being the doers around the world joining in the live session in June. When we have a live session, we will follow the iDOOS principles of collaboration and code of conduct, and we'll talk about this a bit when we get started. And we'll also talk a little bit about how we'll use Zoom in the live presentation. Again, the purpose of a live presentation is to have more time for exchange and using the pulse reactions and such, and using the breakout rooms. So the recording is just kind of for linking to materials. This course is going through the research data lifecycle with an eye towards deep ocean science. And this is module 3, where we're going to be diving into the process, describe, and assure steps. And to recap modules 1 and 2, we first defined FAIR data, findable, accessible, interoperable, and reusable. We learned about the F and the A by doing some homework. And we learned about the steps, but now we're going to actually dive into the steps. We had breakout rooms to discuss good enough practices. Today we'll learn about best practices. And previously we also learned about general tools that we can use to collaborate with each other in the data lifecycle. Today we'll learn more specific tools. What will we learn in module 3? We're going to, again, dive more fully into the steps in the data lifecycle, focusing on practices, tools, and standards in the ocean sciences. And the goal of the module is to be able to reach the level in the Bloom's technology of apply for the describe step. I'd like for you to understand interoperability, the I, and FAIR data. And we'd like to remember concepts related to the process and assure steps of the life cycle. I've been quoting from this resource linked here for all the modules throughout this course on the benefits of FAIR data. And today in module 3, we're focusing on this second quote: to enable easier data integration within and across disciplines. So we're focusing on the I, interoperability of FAIR data today. Interoperability is important in our framework for ocean observing, as introduced in the DOERs data training last year. This is the framework behind the global ocean observing system. And in particular, when we talk about the assembly of data from multiple sources into products, think about how all these data from different instruments and different platforms need to be able to be put together to be in formats and using standards that are interoperable so that you can actually combine the data into products. Or even, analyze the data to make comparisons. The first session of module 3, so we'll have two sessions. They're split into 2 separate hours, 2 separate days, so that we have some homework in between. This first session is split into 4 parts, where we'll start with an activity with the Ocean Best Practices System using our anchoring article by Perlman et al. We'll do a couple of slides to really look into the process step and relating it to the I in FAIR data. The breakout rooms are going to be for discussing tools for the process step. And then we'll do a demo before leading into the homework. First things first is to define what is a best practice. So in our anchoring article by Perlman et al., you'll find there's a section about how the term best practice, it can be used loosely, but we want to have a definition that we follow here in this course. And we'll follow from this paper and from the Ocean Best Practices repository. It's a methodology that has repeatedly produced superior results relative to other methodologies. And it's adopted and employed by multiple organizations. Important in iDOOS, right? Because we have partner organizations from the US and from other countries as well. The Ocean Best Practices System is a repository in which one can deposit an article, a technical report or article related to a best practice, and you can obtain a DOI, Digital Object Identifier, for your report. This repository is co-sponsored by the International Oceanographic Data and Information Exchange. You may have heard of IODE, and it's also co-sponsored by DOERs. So interesting for the doers in the room, there is an early career ocean practices ambassadors program that you might want to look into. So in any case, I noted in their logos that they include for the Ocean Best Practices System, you'll see a lot of the same logos that are used with partners in the DOERs program. And let's highlight, throughout this talk, we'll be highlighting practices implemented in DOERs. And in particular here, you'll see I've circled quite a number of DOERs partners today because this is our world, ocean best practices. So I'll try to hit on quite a number of these different partners when I show examples in the upcoming slides. So in the live session, we're going to search the Ocean Best Practices repository. And I thought, rather than just search for something that I'm interested in, I was listening at the virtual annual meeting in May, and I heard Sergio, one of our plenary speakers, a doer, mention the need for standardized protocols. And I thought, oh, I should ask Sergio what to look for in this OBP system. So contacted over Slack, and Sergio cannot join us during the live session, but has asked us to search for echo sounder processing. So in the live session, we will go to the repository. We'll search for echo sounder processing. We'll actually find that we want to do a Boolean search to narrow our results. And if we'd like to, on your own, you can search for other terms of interest or other topics of interest. And I also point out that at the virtual meeting, the Habitat and Ecological Mapping Workshop also had a bullet point. They talked about the need for standardized and repeatable methodologies to acquire and process data. So this is really related to our collect and process steps, right? So how do you become aware of best practices? This is one way. It's a great resource. It's a more targeted way to find than just doing your, search the web, right? Here is a nice way to target your search. I would like to detail many benefits of using community best practices, but I thought this quote was useful because it really highlights the I in the FAIR data. And it's from a paper by Buck et al. About ocean data product integration. And the quote is, best practices complement standards in supporting improved interoperability and data information exchange. So we need to be aware of best practices. We also should be aware of standards. So the subsequent slides will start to get us aware of standards in our domain. I like to jump into things, so I'm really going to jump into standards in this first session, and then we'll follow up and we'll repeat some of the words, and we'll learn a little bit more in the second session. Okay, some of these terms may be new to you today, and then they'll become more familiar with time in the second session. Standards tend to be associated with file formats. So in our infographic for the research data lifecycle, one of the links that we selected was, it's called Pick a File Format. So the link is here. The link is to a resource that lists many common file formats in the ocean sciences domains. And so I'm only going to be able to highlight a couple in this module. So we'll be focusing only on geospatial raster data and tabular data. But I do want to say that I realize that other data types are important in our domain. And so you can check this resource for suggested file formats to use for geospatial vector data, imagery, sounds, and other data types. In any case, the formats that we will highlight in our DOERs training are NetCDF, and we'll do a particular format for tabular data. NetCDF. In module 2, we had a great exchange in the chat and in the Slack about NetCDF, and it was noticed that some oceanographers, often the physical and environmental oceanographers, are using NetCDF, while the biological oceanographers are tending to use a lot of tabular data. And how do we go in between these formats? So I thought it would be important to just let everybody kind of know a little bit more about each of these separate formats. And then when you get into your breakout rooms, you can talk a little bit more about how you are bridging the world between the two. Learn about each, what's good about each type of format, and then how you work in between. Network Common Data Format, as defined here at the Unidata webpage, is a self-describing, machine-independent data format. It supports array-oriented data, so I learned it initially as for gridded data, but you can also do trajectories, for example. I'll point to a link with different templates that you can use for different kind of types of data if you want to start with a template. I point you to a resource here that has a great introduction to NetCDF, from which I will excerpt just a bit for today's lesson. I thought what's kind of most important to point you to is the templates, and also to let you know that it's used a lot in the geological oceanography as well. So and we'll see where it's used for bathymetric data formats. But I think what I like a lot about NetCDF is that it's three-dimensional, but including time, right? So it's multi-dimensional. It's more than three-dimensional. I usually see it, using 3 dimensions in time. It's a standard delivery format for many repositories in our domain: the Ocean Observatories Initiative, NASA's Physical Oceanography Data Center, the British Oceanography Data Center, and other repositories. So we'll try to do an example with it in module 4 when we reuse data. Again, the breakout rooms will help you to be able to talk more about tools that you're using today. If you join the NetCDF room today, to talk about tools you're using for working with NetCDF files, and then on the second session, you can talk more about how the metadata are incorporated into this self-describing format. So the collect step pointed to file formats, but you don't collect data into NetCDF format, right? It requires some data processing to get the data into that format. So in fact, with many formats, there's some processing required. And so as an example, I'm showing you here from the DOERs Seabed 2030 project. If you go into the GEBCO's website and look at how to contribute your own data to this Seabed 2030, you'll see that you check a box for how processed are your data. Are they raw? Are they processed with some, we'll learn about QA/QC in the second session. Are they gridded? What kind of gridded data products? In this particular GEBCO webpage, you'll see that we go to different dimensions of NetCDF datasets, a raster format, or a GeoTIFF. The processing step may also be involved for tabular data. Don't think you're getting out of the need for processing just because you have beautiful columns of data with wonderful column headers, right? It still takes some work to transform to standardized tabular formats. So in the infographic, there's some really great helpful tips on how to avoid challenges in terms of your data entries in spreadsheets. So there's this formatting problems, but I'll say they're problems that are solved in this particular lesson. You can see kind of ways to make sure that your columns have only one type of information, basically don't have two types of information in the same column, etc. And later in today's slides, during the live session, I'll demo how I am transforming some data to a common format for tabular biodiversity data called Darwin Core. Also, during module 2, there was a little discussion in the chat about the word interoperability, and I think at the time the person who was chatting was thinking that interoperability meant that they could run a data file given their operating system and the packages that they had. So far, I've been talking about interoperability in terms of combining data or working with data, making data products. But it is true, we have to think about interoperability, of course, as data interoperating with software applications and pipelines. So you think about interoperability during the storage process. So that's what we talked about in module 1, and then in processing and in analysis. And there's a great group in ESIP. I've mentioned ESIP several times in modules 1 and 2. It's the Earth Science Information Partners, data professionals. In this case, this link goes to a workshop held by a biological data standards group, but they came up with kind of just the top hits on how to make your data software ready. And we've already covered a couple of non-proprietary formats. This idea of structuring tabular data in long format is, in addition to what I was just talking about in terms of splitting information into multiple columns, this would be essentially choosing how you provide out I don't know, how do I see this so often? I see many times it's kind of like switching between transposing. Like if you have a matrix and you have a lot of columns, you transpose those into rows. This is what I'm talking about in terms of going to a long format. Processing step. I've just talked about transformation from wide to long. And this cartoon is really I think is probably the easiest way to explain all the steps that might be involved between collecting and processing your data. I'll pause in the live session and let people read the cartoon. Reproducibility is, in this case, we're going to think about it in terms of you being able to run the code that you developed earlier, and it'll still work to, go from your unprocessed to processed data. But for reproducibility, you really have to also think about how other people can rerun your code. And there are many publications that define repeatability versus replicability versus reproducibility. But in any case, think about reproducibility as being able to really understand what happened between when your data was raw and when you provide out your product. Reproducibility is a set of best practices. It's not one tool. And in the context of this data training, it's not just that you ultimately could share your code like we talked about in module 1, but it's how you can provide some descriptive metadata to help people understand how to step through the process. So in our infographic, we also link to a video if you want to kind of see an example of going from interacting with spreadsheets to using an open source programming language and scripts for a pipeline for data processing. So I looked back at our Jamboard from module 2, in which we talked about tools that we were using to collaborate on the process step. And we were trying to stay really general, and guess what we did? We were talking about using R, Python, GitHub, GIS in general, but we didn't get into the actual tools, right? So what I'm hoping we can do in the breakout rooms today is we can talk about the actual tools that we are using for particular data sets, and we can learn from each other. Basically, we're not going to go with the Jamboard today. It's already very full, but within your breakout room, this is the time it's okay to talk about the actual tools that you're using. And here is just a smattering of logos from a variety of types of software that we use in our domain, ocean sciences. And in this case, I'm talking not only about process, but also analyzing and visualizing data. And how do you find out about new software that you might want to use? You might read it in a paper, a journal article, or you can learn about it from your peers, which is the goal of our breakout rooms today. In the breakout room, I'm hoping that you feel free to talk about tools you're using, not only to process, but also to analyze and visualize your data, because this taxonomy of skills for data-intensive research, it has skills that we don't have enough time to cover in the FAIR data training. The FAIR data training is really focused so far on data management, collaboration for working on the data. Today, we're looking at processing, but there's a lot of software skills, right? And there's a lot of analysis and visualization that's needed to get information from the data. We don't have as much time in this training to really cover those topics. So please feel free in the breakout room to talk about tools you're using for those. All the breakout rooms will have a particular link here, Tools of the Trade, that has a prompt for discussing factors for selecting the applications that you use for your project. And each breakout room will also have a list of tools to stimulate discussion. So during the live session, we'll have a poll And we'll see how people want to distribute themselves into these 5 different breakout rooms. They're all associated to DOERs working groups that presented at the recent virtual annual meeting. And then after the breakout room, we'll touch base. Just each room will have a seafarer to guide the facility. And we'll see if any tools maybe came up in multiple rooms. That'll be exciting. So as we move from the process step to the describe step, I want to take a pulse poll of the room first and just ask everyone, have you heard of a controlled vocabulary? Maybe. Maybe not. Maybe we've heard of the term data dictionary, thesaurus, ontology, different words. But what we'll do to end the first session is we're going to learn by doing, and I'm going to demonstrate. I'm going to introduce the room to a controlled vocabulary, standard, and format. I'm going to demonstrate the use of Darwin Core. In particular, we'll actually open up the Darwin Core terms, and I'll have my spreadsheet open and I'll show how I selected terms as new column headers. I'm going to use my own data, so I'm not showing it in the recording here. But I'll start with something that we probably all have in our data sets. We probably all have dates in our data sets. So that is for everyone, and then scientific name is for the biologists in the room that might use Darwin Core. The demo is a lead-in to the homework where I will ask anyone, to think about their own data and match a parameter or metadata from, what sampling instrument are you using? What marine region are you studying? We want to match something in your data to a controlled vocabulary. The forum is brief, but it'll provide some, it'll be very interesting to see what people use, and then we can use that to update the slides that you're going to see next here. All right, that'll be the end of the first session. I always like to collect some feedback, and we're going to keep the Zoom open afterwards for additional Q&A questions and also for peer-to-peer networking. Right, so I'll get into the second session now. The second session will start with some of the same slides because I don't expect everyone to be able to attend both the first and the second. It's totally fine to just jump in and join the second session, even if you haven't joined the first. So I'll restate some of our introductory slides. I'd like to recap the first session just by saying we explored the Ocean Best Practices repository. We touched on data formats, but I was pretty brief with NetCDF there, right? So hopefully in the breakout rooms we have more time to get into details. And in the breakout rooms we talked about process tools. So what are we going to learn in the second session? So the second session has 3 parts. It starts with slides and an activity for metadata. And then we'll have our breakout rooms where these will be associated to vocabularies and standards for the describe step. Then we'll have some slides and activity for the assure step before ending the day. Metadata. Metadata answer a lot of questions. We're going to focus on what, where, when, and how. The why, it often goes, if they ask for a description or abstract for your data set, that's usually where the why goes. And the who, we're going to focus on who in the next module, module 4. And again, we're looking at the describe step in our infographic. Here we'll be able to refer to some of the homework results in terms of what were people looking for? What terminology were people trying to match to? So the first control vocabulary that I want to mention is associated to the NetCDF format that I so briefly mentioned in the previous slides. The climate and forecast standard names are often used in the ocean sciences. And I just pointed to a couple of examples here in the physical sciences. The names are more fleshed out for the physical and chemical sciences. For example, seawater density, and it's pointing out an appropriate use as opposed to potential density, which is also really important in deep ocean science. I also wanted to point out that we don't have enough time in our training to get into units, but there are controlled vocabularies for units as well. And that can be important in, you know, so many cases, you have to do conversion of units. So controlled vocabularies, we'll hear about, we'll know how many people out of the previous session had heard of controlled vocabulary, but now we're actually going to go in and we'll search this particular server out of the National Environmental Research Council of UK. This is offered, you can also find it through the British Oceanographic Data Center. And it's a really great way to search for vocabulary terms across collections, inclusive of CF, by the way. I don't think it includes Darwin Core, but I'll check that before I get to class next week. And I had this search as a little tip in the homework in the form, but some of you might not have been able to interact with it. So we'll go through it, and just as we'll do it live, but I also provide an example here in the slides because I wanted to go back to the seawater density from the standard name. And in this particular collection within the NERC vocabularies, what I'm pointing out here is that other vocabularies have broad and narrow terms that help you kind of see how descriptive you need to be about your terms. So sometimes if you just want to say what your data set is about, you might choose a broad term like density of the water column. But if you wanted to like really define the parameter, like the column header, the variable in your data set, you might use a much more narrow term, like here, sigma theta of the water body by thermosalinograph computation using the UNESCO algorithm. Right. And another thing I wanted to point out with the in this particular set of vocabularies is they're using a universal resource identifier that is linked by HTTP, and an identifier that is would allow you to refer back to this phrase. Kind of like it's not really a number, right? It's an address, but it refers to this exact phrase. And it can be versioned as well. So this concept of identifiers is important throughout the metadata. Here, we just talked about an identifier for a vocabulary term. Another slide is going to show you identifiers for scientific names. We probably will see that in the live demo from session 1. Physical samples can have identifiers. We recommend the international geosample numbers linked here. And for people, we will recommend an identifier in module 4. Referring back to the ESIP working session from January earlier this year, make your data software ready. This was one of the tips, was to use globally unique identifiers throughout your metadata. And referring all the way back to module 1, the FAIR principles, the data set will have a persistent identifier if we assign a DOI. So what? Identifiers for taxa, if you're looking at species in your data set, if you're doing biodiversity, one taxonomic reference database that is well used in our community is the World Register of Marine Species, WORMS. And I show here an example from the World Register of Deep Sea Species, or WORDS. Again, this concept of matching to a taxonomic authority is one of the ESIP go-to points. And I should have already demonstrated this in the live session. One. Metadata where. So there's a great resource here about getting your Latin long to decimal degrees. This graphic is nice because it's basically talking about the precision, but there's something missing from the graphic, right? So the graphic is using N and W for north and west, and for decimal degrees, I would recommend, of course, using the negative for the west and the south coordinates. A great vocabulary for regions of the ocean, for also to find vocabularies for subsea names, feature names, is marineregions.org. And again, to make your data software ready, recording in decimal degrees, also referencing a geographic coordinate system can be important. Metadata when. Time can be challenging to work with, especially if you use if you're going between software environments, if you're going between spreadsheet and code. Even today, I was using a NetCDF file in my own work, and I was having some challenges with time. One thing to make it easier is to use Universal Coordinated Time, UTC, or to just be sure to declare your time zone using in the string for the time that you're using. Here is the ISO 8601 standard. Here is how we would declare UTC time using T and Z. I've also seen it without these, but T and Z is more specific. And then declaring a time zone by using, in this case, UTC -7 hours. Make your data software ready, again, says follow the ISO 8601. Metadata how. Now I'll use another phrase and ask how many people in the room have heard of data provenance. Data provenance is the history of your data set. How did it come to be? Describing how you process the data allows users to briefly review your work. So if you were to publish out your data, you have a data package, users can review your work without having to create the local environment and run your processing code. So I've already alluded to packaging data with metadata. And we've talked a little bit about two different formats, NetCDF as a format and Darwin Core as a format. And they're different in the sense that NetCDF combined with the CF standard names is a way to, and I used the word self-describing as well before, it's a way to combine the metadata with the data in the same file. Darwin Core as a format, we haven't even mentioned this particular metadata standard yet, EML, the ecological metadata language. This tabular data often is combined with metadata as separate files. So you have multiple entities to be kind of managing when you're working in this world of Darwin Core as compared to the world of NetCDF. Another format that we can only touch on today, but I want to thank our seafarers, Dawn Wright, for providing a slide for us, is the bathymetric attributed grid format, or BAG. And that is a combined file with data and metadata. And you may have, especially those working with NOAA, the US NOAA, may have heard of the ISO 19115 standard. That is another metadata standard that is often used with tabular data, and also it's used with any kind of data files. It's a separate metadata file, and NOAA has a nice form that steps you through how to provide information for that. What do we mean by having this combination of metadata and data? Here's an actual example from a DOERs partner, the Ocean Observatories Initiative, OOI. And what I've done here is I've actually used a example provided by Colin Dobson, thank you, from the OOI data team. And I literally did this earlier today. I was working with this NetCDF file, and I pull it in, and I'm printing here. There's a lot more metadata that I printed out, but I just kind of excerpted so you can see that it's reporting the use of the standard name seawater temperature for this variable. That over here we plot, ooh, looks like I need to put this plot on top so you can see that we're plotting temperature versus pressure. And this is for a glider that's going to about 1,000 meters depth in the Erminger Sea in the North Atlantic. For Darwin Core terms and the format, pulling these all together, I wanted to give you an example of the package. So this is the Darwin Core archive is what they call it. And it's where they package the tabular data files, text files, with metadata, the EML metadata. And our seafarer, who will be joining us, Maureen Lebrac, attended a recent workshop and contributed a data package to OBIS, but I saw that package was not for deep sea data. So I just went into the US OBIS node, and lo and behold, the most recent data set that was contributed was for a deep sea data set from the Deepwater program. And it's just showing you that in this case, when you download this, DWCA stands for Darwin Core Archive, and you can either grab the whole record, the whole package, or you can just inspect quickly the metadata, the EML file, which will tell you a bit of the what, where, who, when. Again, I want to thank Dawn Wright for helping us with learn a little bit more about the bathymetric attributed grid format, the BAG format, which is like NetCDF in that metadata are included with data into the same product, but it's not in the grid itself. This graphic shows, it's very helpful in showing that you have this three-dimensional data set, but you also have an uncertainty information for every point of the data set. And after we do our breakout rooms, I'll talk a little bit more about that. Also, Don wanted to point out that there are particular tools that you can use to combine your data and metadata into this standard format. And if we didn't get to this tool in one of the working group rooms from session 1, I wanted to make sure people were aware of that. So the breakout rooms for the session 2 are going to dive more into the vocabularies and standards. It's a time for you to, again, discuss the data formats, especially for those of you who wanted to learn more about NetCDF. Perhaps join the NetCDF room. I think everyone should feel free to join whatever room they want to learn more about. Maybe there are some folks in the NetCDF world who want to learn more about the Darwin Core world. Please feel free to join the Darwin Core room. Well, each room will have a seafarer facilitator to help everyone learn more. We'll also have a room for those who have asked for more networking for GIS type work. And we'll also have a room just to explore more fully the vocabularies, because there's so many vocabularies available through that server. After the breakout rooms, we're going to just, touch base, see if any of the rooms, talked about what's happening in the other room, the format from the other room. And I'll also want to find out if any of the rooms touched on the assure step, the next step in the data lifecycle. The assure step actually starts before the collect step. And so I want to thank seafarer Sarah O'Connor for this really nice, just very clear description to kind of to differentiate quality assurance from quality control. Assurance is defect prevention. You do it before collecting the data and while. And control is defect detection after the data are collected. Also, our infographic points to this really wonderful paper, Bushnell et al., 2019, which really talks you through all the different things that you can do for quality assurance before and during data collection. What can you do for quality control? So after you already have your data in hand, I've organized this by the metadata. So what and how, where and when. So if you collect your data with instruments, the number one, and this came up just earlier for me this week too, just the first thing I do is check that the values are sensible, right? Within the range of the instrument and the property that you're measuring. And if you're getting analytical results from a lab, you check that the values are within the detection limit. Check that they're valid for what you expect, for what's being measured. For the where and the when, I usually, oftentimes for metadata, you want to provide the bounding box, the limits, for example. And so when you're processing, figure out the limits for your geographic coverage, just check that they make sense. Do a plot. I often do that. I plot where the data points are just to make sure it visually looks correct to me. And when also check the temporal coverage. This just happened today too. There was, a couple of time records got messed up. I don't know why. And like 3 rows of data out of a huge table, but we found those rows because the temporal coverage looked out of the ordinary. So the assure step, of course, continues after the process step. And we gave an example back in module 2 for the Ocean Observatories Initiative. We talked about how quality, but we didn't use the terms at the time, right? So now you know it's quality assurance for reviewing the calibration metadata, and then quality control for putting flags on the process data. So flags can be used to indicate the quality of data. There are many flagging schemes, but the one that I really wanted all of us to know about is the UNESCO or IODE primary level quality flags. I link you to the document, you see the values. So let's just step through this because these flags are adopted, and I'll show you on the next slide, by the core TOD flagging system, which is used by some of our DOERs partners. 1 is good data, 2, not evaluated, not known, 3, questionable, suspect, we'll find out, might be interesting, 4 is bad, 9 missing data. So this is showing how core TOD, or the quality assurance, quality control of real-time oceanographic data, core TOD, has adopted those IODE flags, but they, two things. Core TOD does not recommend the use of 2 not evaluated. Why? Because one of their best practices is to always evaluate every data point. But they highlight that for flag 3, it's not necessarily questionable or suspect, but it could actually be data of high interest. So that's what I'm going to point out in our example for DOERs. I have a fully fleshed out demo that we can step through to look at core TOD flags for some deep ocean data from the Argentine basin in the Southern Atlantic Ocean. And I fully provide an example here through this link, but we'll just be able to step through one Data Explorer view in the live session. Also in the live session, I'm hoping to talk a little bit about a quality control process that's going on within ocean sites, which is another DOERs partner. And it's very unique to deep ocean science, right? So what do we have to think about that coral reef researchers or anyone working in coastal systems won't? we have to think about pressure. And pressure can have an effect on our sensors. And so it's something that, just like I was saying, what can you do to ensure quality control? If you're working with very deep data, there are some things that you want to think about with regard to the effect of high pressure on your sensor. The parting thought, the last thing, is that I think this is going to become more and more important in the future, is not only doing data quality flags, but also providing information about data uncertainty. And this has already come up in the earlier slide from our seafarer Don Wright for the bag format. I think this is a really nice visual explanation of data uncertainty from the International Hydrographic Organization's bathymetry cookbook. And it's showing you kind of the difference between what you would expect for true depth, and then what happens if your data are not precise. Sorry, not precise, not accurate is up here. Here's precise and accurate. Here's precise and accurate. Where's our not accurate, not precise? If you work with bathymetry data, this happens even if it's just echo sounding data. This does not in any way mean the data quality are bad or suspect, right? It literally could be the data are being received just fine, but the uncertainty is happening because of how the ship is moving, how the bottom surface is changing, how how all the different sensing is going on, the different water masses in the water column affecting the acoustic travel time of the sound, right? So in the slides, there's also a link to a presentation at an ESIP meeting, which is more a climate model example of how you can provide uncertainty information per data point for observational data important for physical oceanography. So at the end of session 2, we'll ask for how did we do? Do you feel you can apply the described step? Do you understand interoperability? Do you feel comfortable about the process and assure steps? To lead into our module 4, here our infographic is just beautiful, right? It goes from collect to process to describe, but you have to consider the describe step early and often. So every step actually has describe. And we learned today that even the assure step starts before collect. So our data cycle has a lot of iteration in it. We'll end with some resources to learn more. This is obviously very selected. I've only listed 3 things here out of what could be probably 100. And I want to acknowledge funding from the National Science Foundation, and some of the slides that were adopted or used today are based on a workshop series developed at the Woods Hole Oceanographic Institution. And we'll be publishing these materials out with a Creative Commons attribution license. And thank you.